What if fixing a genetic disease was as simple as correcting a typo? Find the mistake in your DNA, delete it, replace it with the correct sequence. That's no longer science fiction. With technologies like CRISPR, scientists have already begun editing the human genome. But now, we've entered gene editing 2.0, a new era that goes far beyond cutting DNA. Today we'll explore how CRISPR works, what's wrong with the original version, and how new tools like base editing and prime editing are changing the future of medicine, agriculture, and biotechnology. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Despite the complicated name, the idea is elegant. It's a natural bacterial immune system. When viruses attack bacteria, they store fragments of viral DNA. Later, they use a protein called Cas9 to recognize and cut that viral DNA. In 2012, scientists including Jennifer Doudna and Emmanuel Charpentier showed that this system could be repurposed to edit genes in almost any organism, a discovery that later earned them the Nobel Prize. CRISPR-Cas9 works like molecular scissors. A guide RNA finds the exact DNA sequence. Cas9 cuts the DNA. The cell repairs the break, sometimes introducing a change. For the first time in history, gene editing became fast, affordable, highly targeted, widely accessible. It revolutionized genome editing technology, but it wasn't perfect. The original CRISPR method relies on making a double-strand break in DNA, and that's where problems begin. When DNA is cut, cells repair it, but that repair process can be unpredictable. It may introduce unintended mutations. There are three main challenges. 1. Off-target effects. CRISPR sometimes cuts similar DNA sequences elsewhere in the genome. 2. Unpredictable repairs. The cell's repair machinery can create insertions or deletions. 3. Limited precision. CRISPR is great at disabling genes, but not ideal for precise single-letter corrections. And since many genetic diseases are caused by just one-letter mistake, scientists needed something more accurate. That's where gene editing 2.0 comes in. Base editing is like using a pencil instead of scissors. Instead of cutting both strands of DNA, base editors chemically convert one DNA letter into another. For example, C to T or A to G, no double-strand break required. This dramatically reduces unintended damage and increases precision. Since many inherited diseases are caused by single-letter mutations, base editing opens the door to correcting thousands of genetic conditions, including certain forms of sickle cell disease, cystic fibrosis, and inherited blindness. This is a major step forward in gene therapy. If base editing is a pencil, prime editing is a word processor. Developed in 2019, prime editing allows scientists to insert new DNA, delete sequences, replace longer stretches, precisely correct mutations. And it does this without fully breaking the DNA strand. Prime editing uses a modified Cas enzyme linked to a reverse transcriptase, essentially writing new genetic instructions directly into the genome. Researchers have described it as search and replace for DNA. This makes genome editing more flexible, more precise, and potentially safer. CRISPR-based therapies have been approved in some countries to treat sickle cell disease. Scientists are also engineering immune cells to fight cancer and exploring treatments for rare genetic disorders. Agriculture gene editing is helping create crops that are drought-resistant, disease-resistant, more nutritious. Unlike traditional GMOs, some gene-edited crops do not introduce foreign DNA. Disease prevention researchers are studying gene drives using CRISPR to reduce malaria transmission by altering mosquito populations. Gene editing 2.0 isn't just theory, It's actively reshaping biotechnology. With great power comes serious responsibility. In 2018, a scientist announced the birth of CRISPR-edited babies, sparking global backlash and ethical debate. Key questions remain. Should we edit human embryos? Where is the line between therapy and enhancement? Who controls access to gene editing technology? As precision improves, so does the urgency of regulation and oversight. Gene editing 2.0 makes changes safer, but also more powerful. CRISPR was just the beginning. Scientists are now exploring RNA editing, temporary gene changes, epigenome editing, turning genes on or off without altering DNA. New CRISPR systems beyond Cas9. Biology is becoming programmable. The future of genetic engineering may include personalized gene therapies, climate-resilient crops, and treatments for diseases we once thought incurable. The question is no longer, can we edit genes? It's, how responsibly and how wisely will we use this power? Gene editing 2.0 is here, and it may redefine medicine, biotechnology, and even what it means to be human. Thanks for watching Astrotech. If you found this video helpful, don't forget to like, share, and subscribe for more amazing tech content. See you in the next video. Stay curious, stay innovative.